Hi, my name is Nikos Petrellis from the Embedded System Design and Applications Laboratory of Electrical and Computer Engineering Department of the University of Peloponnese, Greece, and the paper I'm going to present is entitled Training Ensemble of Regression Trees Models for Fish Morphology Measurement. It has been conducted in the context of the Smart Fish project funded by the Hellenic Fund Research Institute. The aim is to measure fish morphological features, for example, skeleton malformations, the dimensions of fish, length projected to weight, to distinguish fish species variations, the localization of body parts for color analysis and estimation of freshness, health conditions of the fish, etc. And the morphological fish features can be determined by the distance and position of landmarks that are used to represent the fish shape. Here we see the positions, the appropriate positions for placing landmarks at fish contour. And the method we have used for shape alignment is a machine learning method called ensemble of regression trees. Each ERT model consists of a number of cascade stages. The default is 10. Each cascade stage has a number of regression trees, binary regression trees. The default is 500 trees per cascade stage. And all binary regression trees have constant length. Constant depth, sorry. The default is 5, so each binary tree has 31 nodes. In each tree node, the gray intensity of a pair of pixels is compared around the landmark. According to the comparison, the next node to be visited is decided, and each node in the regression tree contains a landmark coordinate correction factor, and this method is based in the paper by Kazemi and Sullivan that you see in this slide. Here is an example. If the red bullet is, for example, the initial landmark position in the ERT model, placed there as an average position of all the photographs, the annotated photographs that were used for training, and the blue bullet is the correct position where the mouth of the fish is denoted, then the correction shown by the arrow is needed. Here in this slide, we see if we traverse, for example, a depth equal to 3 binary regression tree and visit 3 of its nodes from the root down to the leaf, how the corrections of the landmark position could be performed. Here are two photographs showing the annotations in the two datasets that we have used. On the left, we see 26 landmarks, while on the right, we see the use of 4 landmarks. Basically, they're used in this case for measuring the length of the fish and its height. The datasets used. The UVMF dataset is available in the Kaggle repository and is constructed by us. There, the photographs, we used 500 OpenSea underwater images from this dataset, and we annotated 26 landmarks as shown earlier, while we used 1000 underwater photographs in order to train landmarks with only 4 landmarks. The images were taken by the experimental setup of the University of Patras, our colleagues in the project. The ERT models trained with 26 landmarks are the ones shown in this slide. You see that they differ in the number of cascade stages, or the trees per cascade stage, or the depth of the trees. And the best normalized error is achieved by A1 model, which consists of a large number of trees per cascade stage. At the bottom of the slide, you can see a fish with the annotated landmarks in a test photograph. Here is also the list of the models that we have trained with only 4 landmarks. We can see again differences in the average error and the worst error according to the parameters of the model selected. And now concerning the frame processing latency, we can say that the software implementation depends on the platform, whether it is x64 or an ARM architecture. The resolution of the frame, the latency is proportional to the resolution of the frame that is analyzed, and thus several seconds may be needed to process just a single image. But with the hardware acceleration support that we have implemented on an AMD Xilinx ZCU 102 FPGA development system, the models can be implemented in hardware only if the following restrictions hold. The tree depth cannot be higher than 6, so no more than 63 nodes per regression tree. The number of trees per cascade stage should not be higher than 600. Otherwise, a larger FPGA could be used to implement models with higher dimensions. In this case, if for example we see the frame processing latency for model A0, it is less than 15 milliseconds. The models with 4 landmarks showed a worse error despite the fact that they were trained in a controlled environment with low number of landmarks. One explanation is that it is difficult to define the precise position for the caudal fin, the upper and the lower landmarks, since the fins are not very clear in the low resolution photographs that we have used, because we wanted to stress the whole system and work at the limits. Larger number of landmarks introduces somehow correlation, and this is maybe the reason why a landmark cannot be found in extremely larger position with its neighboring ones. The models trained with 26 landmarks achieved lower error in the photographs taken from the aquarium and used with the 4 landmark models, while hardware acceleration boosts the ERT model performance, since a single fish shape alignment latency can be performed in less than 15 milliseconds. Now, concerning our future work, it will focus on the integration of this shape alignment method with fish tracking, as well as other applications such as the customer mobile application, conveyor belt monitoring at the hatchery, as well as adding some cloud services. We also plan to extend our testing in real-time conditions, and finally to model the effect of other parameters, like for example the borders in the image patches that we extract from the initial video frames, the resolution of the photographs, as well as the lighting conditions. Thank you very much for your attention.